Hello everyone. Have you ever thought about the fact that nutrients are synthesized at the top of the tree, while water and minerals are absorbed from the bottom of the tree? So my question is, how are the nutrients from the top transported down to the bottom, and how are the water and minerals from the bottom transported to the top? Well, to solve that question, join me in BioWorld. So far, in our STPM syllabus, I've discussed epidermal tissues in my specialized plant cells video part 1 and ground tissues in my specialized plant cells video part 2. So today it's going to be all about vascular tissues. So come join me. Here is a cross-section of the stem as seen under the microscope. The outermost layer is made up of the epidermis, that is the dermal tissue. The cortex layer is made up of the ground tissue, which includes large cells like the parenchyma and smaller cells like the collenchyma and sclerenchyma. What's left are the vascular tissues. The vascular tissues are found in a circular structure known as the vascular bundle. There are 3 layers in the vascular bundle. The layer below, the xylem, the layer above, the phloem, and the layer in between is actually the meristematic tissue, that is the vascular cambium. But our focus is primarily on the xylem and the phloem that make up the vascular tissue. As you can see, this is a microscope image of the longitudinal section of the stem, where you can notice that both xylem and phloem are elongated cells. My focus now will be on xylem. Xylem tissues are made up of 4 different cells: the xylem parenchyma, the xylem fibers, the xylem vessels, and the tracheids. Xylem parenchyma are just like the parenchyma of ground tissue. It has a thin cell wall made of cellulose. It has nucleus, cytoplasm, and vacuole. So xylem parenchyma are living cells that carry out metabolic processes. The xylem fibers are just like the sclerenchyma in ground tissue. It has a thick cell wall with lignin deposits. So xylem fibers are dead cells with empty lumen. However, xylem fibers cannot transport water and minerals since the lumen is too narrow. Instead, xylem fibers are suitable for support in plants. The cells that are actually responsible for transport of water and minerals are xylem vessels and tracheids. The similarity between these two cells are in their structure. Both cells are long and tube-like, suitable to transport water and minerals from the root right up to the shoot. The cell wall of both these cells are lignified, so because of that, both the cells are dead cells. They have no cytoplasm. Their center is a hollow lumen suitable for transport without obstruction. However, from the diagram, you can notice the difference between xylem vessels and tracheids. For one, the diameter of the xylem vessels is larger than the diameter of the trachea. This indicates that transport is more efficient in the vessels compared to the tracheids. Besides that, the xylem vessels are actually a number of individual cells that have fused together. Each individual cell in the xylem vessel is called a vessel element. These vessel elements fuse together so that the cell wall disappears. Once the cell wall disappears, these xylem vessels have open ends that enable transport of water and minerals without obstruction. However, tracheids are individual cells. Their structure has a tapering end where you can see it is pointed at the tips here. And since these individual tracheid cells are not fused together, they still have a cell wall. But to enable the transport of the water and minerals from cell to cell, there are tiny holes in the cell wall called pit. To learn more about the transport of water in xylem, you can check out my video on water transport as well as water uptake. These pretty patterns are actually lignification patterns found on xylem vessels and tracheids. Lignification of the cell wall not only enables the cell to carry out transport of water and minerals, but also enables the cells to carry out support. Let me introduce each type of pattern. The pattern with the least amount of lignification is called annular lignification. And when a little bit more of lignin is added, it is called spiral lignification. But when larger amounts of lignin is added into the cell wall, we have the reticulate or scalariformal lignification, and the maximum amount of lignin will produce pitted lignification. Xylem cells that have annular and spiral lignification are called protoxylems. These type of xylem tissues are able to elongate. So, young plants usually will have protoxylem with annular and spiral lignification so that the xylem can elongate with the growth of the young plant. The xylems with harder or larger amounts of lignification are called metaxylems. These type of xylems have become hard and will not be able to elongate, so they are more suitable for support in adult plants. So now we know all about the xylem. Let's move on to discuss the phloem. This is the cross section of the phloem. And just like the xylem tissue, phloem tissue is also made up of a number of cells. We'll start first with these large cells. These are the phloem parenchyma that are similar to the parenchyma in ground tissues. Then we look at the cells here that show a much thicker cell wall. These include the phloem fibers and phloem sclerids. They are similar to the sclerenchyma in ground tissues. Over here, you have some large cells and some tiny cells. These two cells make up the phloem tissue that is involved in transport. Here is a longitudinal section of the smaller cell and the larger cell. The larger cell is known as the sieve tube. The sieve tubes are joined together at the sieve plate, where you find there are tiny little holes here called the sieve pore. Transport of nutrient occurs along this passage. Beside the sieve tube, there is the companion cell. The sieve tube is a long tube because it is a fusion of many individual sieve tube cells called the sieve tube element. The cell wall between the sieve tube elements fuse together to form the sieve plate. To enable transport from sieve tube element to another sieve tube element, there are tiny holes in the sieve plate called the sieve pore. Although the center of the sieve tube is empty, the sieve tube strangely is a living cell. It has no nucleus and its cytoplasm is limited to the periphery of the cell. So how is it possible that the sieve tube remains alive? This is because sieve tubes have what is known as a companion cell. These companion cells are active living cells with dense cytoplasm. The companion cell and sieve tube work together to carry out translocation. Translocation comes from the word transfer location. So, organic molecules produced by photosynthesizing leaves can be transferred from the leaves above to the roots below, or it can be transferred from the leaves below to, say, the fruits above. So translocation is a two-way transport, either up to down or down to up. Now the sieve tube provides enough space for this translocation to occur since the sieve tube has a very thin layer of cytoplasm which is pushed to the periphery of the cell. The sieve plates provide space for the organic molecules to be transferred between the sieve tube elements through the sieve pore. Of course, the transport would require ATP, which is provided by the companion cell. You can learn more about the translocation process through my two videos on translocation in phloem. So with that, I conclude our discussion on specialized plant cells. Oh wait, I'll meet you in my next video in specialized animal cells. Bye-bye.